At Sokoine University, I pursued my PhD. I majored on viral magic fever. Speaking of viruses that cause bleeding, hemorrhage, for example, Ebola, Marburg, Rift Valley. Those are the main disease and pathogen that I dealt with during my PhD studies. Previously, we had specific diseases for humans and for animals, but again, as we are coming up, we see their interactions. So I saw the need of us doctors, medical people, also to know a lot about other diseases that are emerging and re-emerging. And my main area of interest was to strengthen capacity for improving this detection and identification of these viruses. I used to hear that the process of PhD is hard, but for me it was a bit not complex because the type of sponsorship that I got. I was sponsored by SACIDS and they would take care of everything. Yeah, they would ensure that your fees are well paid, you have place to live, you have facilities to conduct your work, you have better mentorship.
There is a disease called Newcastle disease. Today, if you go to any village in sub-Saharan Africa and you talk about Newcastle disease, anyone knows this disease. And the only way to fight against this disease is by using vaccine. And it's happened that this virus has about 21 genotypes already identified. And the vaccine we are using until now is developed by the genotype 2 of the said virus. My first goal in my work was firstly to know which genotype we have actually in Tanzania, because farmers are complaining that the vaccine they are using, but they still have a problem with the disease. So firstly, we did collect sample from outbreak of this disease in different regions of Tanzania, and we realized that actually the genotype which is in Tanzania is the genotype 7. And the said genotype 7 is the most virulent genotype of the virus. So what we are trying to do now is to propose a new vaccine developed using the genotype 7, which is the most virulent, and use it to fight against this disease in Tanzania, and why not in Africa.
I did my research on TB, specifically in illicit drug users. I did prevalence of TB and risk factors among illicit drug users in Temeke, Dar es Salaam, within Tanzania. Several medications and even vaccines like BCG have been developed, but to date, TB is still a problem. So in order to solve these kind of issues, we have to look on populations at high risk, because this population at high risk may act as reservoir to general population. I decided to work on illicit drug users because they are among high risk populations due to their lifestyles and the behavioral changes due to illicit drug users. Apart from having knowledge, but also they lack ability to go to the healthcare professions. I intended to give them the ability to have early diagnosis. I went to their specific hot spots area, but also guiding some members from their own so that they could take sputum samples and send them to the hospitals.
I'm actually doing my research on the epidemiology and transmission dynamics of brucella or brucellosis in South Sudan and Tanzania. So I'll be looking into the prevalence. I wanted to determine the prevalence of the disease across various species: cattle, sheep, goats, and as well human. And also I will do culture. I will be culturing the bacteria and get an isolate and extract DNA so that I will be able to do molecular characterization of the Brucella, because there are various strains of Brucella. So I wanted to know what is actually the circulating Brucella species in South Sudan and as well in Tanzania. So with my study, I will be able to know which species or which strain is actually that causes problem in human or in animals. So can a cattle be infected with the strains from the goats? So looking into that will give us a better picture towards the control and prevention of this disease.
My research is basically about the epidemiology of livestock diseases in sub-Saharan Africa. Specifically, I'm looking at the bovine tuberculosis as a livestock disease. So the problem that I'm trying to solve is that later I'll create like a risk map in trying to identify areas with high risk and those areas with low risk. And for those areas that have high risk, probably they can intervene in terms of control of the disease.
Within the students and candidates that we have trained, if I compare from when we started and now, I'm very pleased that we have really developed systems and infrastructure in place and built that capacity to be able to address infectious diseases in the settings of Africa.